welcome to this explainer. If you've ever wondered exactly how your body so perfectly regulates your metabolism, you are about to get a front row seat to the microscopic machinery that makes it all happen. Today, we're diving deep into the human body to explore what is, quite literally, a fascinating biological manufacturing plant.
Think of the thyrocyte as a microscopic factory located right within your thyroid gland. Its entire existence, its sole purpose, is dedicated to sustaining your systemic health by processing just one single essential atom, iodine. It's an absolute masterpiece of biological engineering.
So here's the roadmap for our factory tour today. We'll start with the iodine supply chain, move on to the thyrocyte factory floor, look at built-in autoregulation, tackle toxins and genetic roadblocks, and finally, we'll wrap up with the medical hijack.
All right, let's jump right into Section 1, the iodine supply chain.
There is an incredibly narrow, almost finicky, Goldilocks zone for dietary iodine. On the extreme low end, a severe deficiency leads to devastating, irreversible cognitive impairment known as cretinism. But if you swing too far in the exact opposite direction, an excessive flood of iodine can actually trigger transient hyperthyroidism. So this supply chain has to be meticulously managed.
To really grasp how your body manages this, imagine following a single iodine atom from your stomach straight down into your systemic circulation. Normally, a healthy thyroid factory extracts maybe 10 to 30% of the supply pool. And this adaptability is just wild. If your body experiences a chronic shortage, that adaptive trapping capacity cranks up to well over 80%. Your thyroid basically goes into overdrive to capture every single atom it can find.
Moving right along, Section 2, the thyrocyte factory floor.
Okay, so the crucial point to understand here is that the thyrocyte has a brilliantly polarized architecture. It's essentially a room with two completely different doors. On one side, you have the basolateral loading dock, which faces your bloodstream and is designed for one job, extracting iodide. On the other side, you've got the apical domain facing the inner follicular lumen, which handles the final assembly of hormones.
Let's zoom in on that loading dock for a second. To drag iodide inside, the factory uses the NIS basolateral door. Now, this isn't just an open gateway. It's a highly active transport machine. It relies on a super strict stoichiometry, basically a very specific ratio, of 2 sodium ions to pull in just 1 iodide ion completely against the concentration gradient. It's kind of like a microscopic water wheel, using the flow of sodium to drag iodine uphill into the cell.
But here's the thing. Getting those NIS doors installed correctly on the cell membrane is no small feat. It requires a massively complex logistics network. AP1A and AP1B complexes act like biological delivery trucks, routing the NIS machinery from the trans-Golgi network straight to the basolateral membrane. If this targeting fails, well, the machinery just piles up uselessly inside the cell.
Let's move to the other side of the cell. Once the iodide is safely inside, it has to be pushed out into the follicular lumen. This is handled by a highly cooperative, redundant network of apical transporters. You've got pendrin, SLC26A7, and ANO1 acting together as a specialized toolkit to make absolutely sure the iodide gets exactly where it needs to go for final assembly.
Once it's out there, the raw iodide hits the organification assembly line, undergoing a really precise 3-step biochemical makeover: oxidation, iodation, and finally, coupling.
Now, you might be wondering, why on earth does this assembly happen completely outside the main cell body? Well, the first step, oxidation, requires hydrogen peroxide, which is extremely reactive. So by running this assembly line outside the cell in the lumen, the thyrocyte protects its own interior from toxic oxidative damage. Out there, the thyroid peroxide enzyme safely constructs your final T3 and T4 metabolic hormones on a massive thyroglobulin scaffold. Talk about brilliant engineering.
And get this, the factory is also incredibly eco-friendly. It relies on an internal recycling loop where an enzyme called DHHAL1 acts as a microscopic salvage yard. It literally strips iodine off inactive precursors to reclaim free iodide. This ensures the manufacturing machinery maintains incredibly high efficiency and doesn't waste a single precious drop of its raw material. Next up, Section 3, built-in autoregulation. So what happens if this factory gets hit with a massive, dangerous surplus of raw materials? To prevent a total toxic metabolic meltdown, the cell hits the emergency brake, a mechanism called the Wolff-Chaikov effect. And the coolest part? It doesn't even wait for signals from the brain. It is an intrinsic, built-in reflex that instantly halts hormone synthesis the second iodine levels spike dangerously high.
When you map this out, you see that during an iodine flood,
thyroid hormone synthesis just drops off a cliff. But the factory can't stay shut down forever, right? So about 24 to 48 hours later, the cell triggers an adaptive escape by forcibly restricting those basolateral NIS doors we talked about earlier. By essentially turning away new deliveries, the intracellular iodine drops and hormone synthesis recovers.
This mechanical perfection is astounding, but it begs a really crucial question regarding the system's vulnerabilities. What happens when this highly specialized factory gets hacked by environmental toxins or, say, a broken genetic code?
Let's dig into that in Section 4, Toxins and Genetic Roadblocks.
Time to look at industrial sabotage, which is ultimately what leads to systemic disease. Because the NIS trap on the basolateral membrane is aggressively designed to grab anything that even remotely looks like iodine, it is incredibly easy to jam. Structural mimics, like perchlorate found in cigarette smoke, can literally wedge into the doors and block them. Alternatively, the factory can be completely overwhelmed by iodine-dense medications like amiodarone, delivering massive payloads that trigger severe system overrides.
And actually, sometimes the blueprints themselves are just flawed from the very beginning. Biallelic mutations in genes like SLC5A5 can cause a total loss of trapping ability. The loss of just one crucial hydroxyl group prevents the machine from coordinating with sodium. Without that grip, the basolateral door simply doesn't work, meaning the factory is entirely broken before it can even start production.
Finally, let's look at Section 5, the medical hijack.
We're going to conclude by seeing how modern medicine actually uses the factory's own machinery against it. Doctors deploy a literal clinical radioisotope arsenal. Because the thyroid is so uniquely greedy for iodine, endocrinologists can sneak radioactive Trojan horses right through the factory doors. Take iodine-131, for example. It emits beta radiation. When the thyroid eagerly traps it, thinking it's food, that radiation provides targeted internal ablation to destroy dangerous tumors.
This dynamic elegantly summarizes the core paradox of the thyrocyte. On one hand, the exact same aggressive, non-discriminating trapping mechanism makes the thyroid terrifyingly vulnerable to environmental toxins and nuclear fallout. Yet, on the other hand, that exact same vulnerability is what allows doctors to precisely deliver life-saving treatments like radioiodine therapy straight to the source.
Humanity literally turned an evolutionary flaw into a medical triumph. And honestly, it leaves us with an incredible question to ponder. If we can hack this microscopic factory to cure thyroid cancer, what other hidden biological vulnerabilities in our bodies are just waiting to be weaponized for healing? Thanks for joining me on this explainer today. Keep learning and stay curious.